There's so much planning is going on at every level. And I thought this is a good time to kind of talk a little bit about the importance of the schools of nursing and what is being done at the schools of nursing. So you as faculty have an idea about what is happening with this, and many of you may have other information that you have gained or gleaned from your local health department or other contacts. But I want to make sure that that you have some information, and then we can begin to, as a group, give some thought about how nursing may be able to respond to this. So let me share some of my screen so I can go through just a little bit of information. I want to give some background to what is currently happening, and some of it I think will be important now, and some that you may want to tuck away for consideration in the future. But I think there are several issues, four main issues that nurses will drive in the mass vaccination effort as the COVID-19 vaccines come to approval. First is going to be knowledge about safety and effectiveness of the vaccine, and be able to do what we do best, and that is personalize it to our community. So what does this mean to me, the nurse, and what does it mean to you, the patient, the family, the community, and so forth? We will also need to be knowledgeable about vaccine handling, management, and administration, because one of the biggest discussions right now involves who is going to give vaccine. And, the first mention is always everyone that's in the Medical Reserve Corps. But the experiences, not only in Kentucky, but across the U.S., are for every 100 people we train in the Medical Reserve Corps, when we have a disaster, we can count on about 2 or 3 to actually be available, unencumbered and unrestricted. So that's not always the answer. And we have done projects in the past demonstrating that nursing students are an untapped resource. They are knowledgeable, they are oftentimes motivated and very humanitarian from the outset, and wanting to be involved in community activities. And then they have the basic core competencies that are important for community response. We will also be driving patient, family, and community education. And this is vital because nurses have within our power to change the perspective in the community from vaccine hesitance to vaccine confidence. And if we do a good job with understanding the data and understanding what is happening, then we literally will be determining whether or not communities accept the vaccine and whether we can stop disease at some level. We will also be very involved and important in passive surveillance processes. That is, keeping an eye out what is happening in the population with respect to vaccine adverse events, and the portion of this that goes on as part of kind of that post market or that phase 4 type of evaluation. And I'll touch on that a bit. I want to just kind of set the stage to say why healthcare workers will be included in phase 1A. This means healthcare workers that have active involvement with patients, or that either are ill or in situations where infection can occur. And it starts at the very beginning. Sure. On the data that we initially had from China that showed of 138 hospitalized patients, 29% of them were healthcare workers. So we knew right at the very beginning that healthcare workers become ill. And now this was, of course, before we really had any idea about transmission and the appropriate use of personal protective equipment. But despite that, we continue to see disease in nurses that exceeds the rate that we identify in the community. We've seen this in hospitalized patients, and we've seen this in long-term care. So we know that there are going to be some specific areas of interest for vaccination. And I'm not even going to touch today on how prioritization will occur, but that's something to keep in the back of your mind, because in the event, just in Jefferson County, for example, we've estimated that there are close to 100,000 in the population of about 800,000 who will define themselves as being a worker in some aspect of healthcare. Now, clearly not all those are frontline healthcare workers, but if we have 10,000 doses of vaccine released early, how will the prioritization occur for those 10,000 doses? So you can begin to see, a lot of the depth of planning and the huge lift that this is going to be in public health. We also recognize that this is a disease nobody wants. For many people, it may be a disease that they get over after 10 days, but for many people, there are prolonged effects to infection. We have individuals that still, after 5 months or 6 months after infection, still have overwhelming fatigue. We have cognitive impairment, we have cardiac issues, we have metabolic issues. So this is a disease you don't want. So we need to be focused on preventing, especially among those who, if they become ill, may suffer more severe consequences, including those that are catastrophic. Well, one of the things, this is just a cartoon that just gives a little bit about how infection occurs, what's happening when we become infected. And this is important for us because it gives an idea so that we begin to understand the vaccines, because all these vaccines are new. Some are completely new development processes. Some are using newer ways of developing vaccines, but everything is largely untested. But this little cartoon just shows at the top left the virus, and those green balls, that's the spike protein or the S protein that you will read about. That is the target for all of the vaccines. And when we inhale, or when we come in contact with that organism, it attaches very effectively to ACE2 inhibitors in our epithelium. And we know we've got ACE2 inhibitors in multiple, all over our body. And it's not just the lung. This is why this is such a sneaky, clever virus. It can affect then all of our body systems. But once it gets into the cell, and that's endocytosis, it gets into the cell and it releases then RNA to begin that whole translation process to make copies of itself. And then once all of that genetic material is reassembled, it is released then as a new virus. So there are two targets for vaccine. One is, it's called a messenger RNA vaccine, and it disrupts the information that is able to occur so that virus can reassemble itself. Everything focuses on the RNA, and understanding then how to change the recognition of that spike protein so our body recognizes it as something foreign. Once that happens, then we produce antibodies, then those antibodies attach to all of those spikes, all of those. So it's that, in this cartoon, I've made like little red, looks like Pac-Man on those proteins. And that's what keeps it from being able then to attach to the ACE2 receptor. So everything that we're producing is trying to get our body to recognize that spike protein with one of the measures of the vaccine. So when you hear about the S protein or spike protein, that's why it is so important. And that ultimately we want to activate our immune cells. So at every level, B cells for immune response, then developing memory cells so we have durability, and then also the T cells that will actually go out and destroy any virus that is identified. So we're trying to use a brand new method for vaccine development to stimulate completely our immune system, because this virus is able to take advantage of every opportunity for ACE2 receptor attachment. So the history is there have been about 150 vaccines in development. There are 5 that are in active clinical trials in the US, and as of today, there are 3 that are actually in phase 3 trial. So those are the ones that are the closest to release. The other 2 are very quickly behind the other 3. So at any point, we may have multiple vaccines that are all available in the general population, all working differently, and we'll be trying to see which one is best, which one produces the best immune response, and which one has a lasting immune response. When we look at those 3 vaccines, there are some issues that as a nurse we need to be concerned about. And I want you to look at this table in the middle under this number of doses and handling. The Pfizer vaccine is the one that is the closest to release, I think, if I understand the material. This a vaccine that, as you're reading information and talking about this cold chain, this is a vial that is delivered that has to remain at minus 70 degrees centigrade until it's removed and put into a refrigerator. And once it's in a refrigerated environment, it's good for 5 days. It has to be diluted. And when it's diluted, each multi-dose vial will have 5 doses. There is no preservative, and the dose is 0.3 cc's. So we will be working with a preservative-free vaccine that's handled, that you have to handle very quickly. You can't let it sit. You are diluting it, again, with no preservative, so we have contamination issues. And we're going to be administering it in a way, in a dose that is unfamiliar. Usually vaccines are 1 cc or half cc. This is 0.3. Not only that, we'll have to use 2 different syringes because of the diluting. We have to have a 3 cc syringe. Administering, we have to have a 1 cc syringe. In addition, we're going to have to make sure that the vaccine is intramuscular, because one of the case reports as part of the vaccine development showed that the individual had vaccine administered, and it was administered subcutaneously, and they had mild tissue necrosis. So that means we have to make sure that we know how to give an IM injection. Now, for many of you that are of a normal BMI, and I don't like to call myself obese, I tell people I prefer nutritionally advantaged, but I'm somebody that I don't get to use because I've, I like ice cream. And because of that, now I need an 1.5-inch needle. So we will have to make sure our vaccine administrator, those that are vaccinators, know how to select the right size needle, as well as all of these procedures. In addition, at least 3 of the vaccines we think will require 2 doses. There is a 4th vaccine that I think will be entering phase 3 trials shortly, the Johnson & Johnson and Janssen vaccine. They're trying to look at that in the provision of only one dose. But what the laboratories are showing now, that the first dose is like a primer. The second dose is the booster that gives us the longer term durability immunity, because we don't want a vaccine that only lasts 6 months. We want a vaccine that is going to last at least a year, in case we have to have this be an annual vaccine, just like a flu shot. So you can see there's a lot to know. In addition, if you'll look, 2 of these are messenger RNA vaccines, which means there's no live component. The AstraZeneca vaccine and the Johnson & Johnson Janssen vaccine both use a live virus vector. The AstraZeneca is the one that they had to stop the trial because someone had the transverse myelitis. That is an autoimmune disorder that may be related to the fact they were using then a live virus to carry the message into the body, so that the body will not recognize the carrier, but will recognize then the antigen to begin to produce antibodies against that spike protein. So you can tell when we talk with our population, many people push back from a live vaccine. So here we have a brand new virus with a vaccine that, even though we're not giving them live coronavirus, it is still a live vaccine. So we're going to have to then be on game as we begin to not only talk to our colleagues in healthcare, but talk to our patients. We'll also have to let people know about the process for getting a vaccine through a clinical trial. And this is just a cartoon just to let you know the processes for even developing something that can be tested. Once there are a variety of pathways for this to go, and this is not our current vaccines are the orange blocks. That's how we currently produce vaccines. We've got the particle and then we adapt it. Now we're looking at genetic engineering for a vaccine. And then once it is in a solution, it has to be tested in the lab. If it doesn't kill lab animals and it produces effect, then it begins to get into safety and then the immune response trials. Phase 1 is usually like 10, 20 people, healthy, all healthy young people, to make sure that it's safe and they produce antibodies. Phase 2, you get 100 or so, maybe more, but you're just looking to make sure that the immune response is strong and that the dose is correct. Phase 3 is where you have larger, usually tens of thousands of people, where you want to make sure that there's no other safety issues, and then again watching the immune response to make sure that there's some continued immunity. This is where you also have the placebos. So half get the vaccine, half get the placebo. And then once you meet the markers for determining the likely effectiveness, that's when it is released. Where the companies are now, because of Operation Warp Speed, already the companies are, as part of money they've received from the federal government, they have stockpiles and are producing the vaccine in the millions of doses. That sounds great, but think we have about 350 million people in the United States. If there are 3 million doses of vaccine, that's not very much. So that's why we're talking about then the questions about how do we prioritize when that happens. So as we talk to the population, we will need to have an understanding about these clinical trials and assuring then not only our colleagues, but the public about the safety measures that are in place to constantly evaluate, and that's what is done in a phase 3 trial. So the vaccines right now that are in phase 3 are looking to make sure that there are no safety surprises. And right now, the messenger RNA vaccines, the only reports that they have had for adverse events have been the ones that we typically see with the vaccine, and that includes things like fever and sore arm. The carrier, the vector vaccine, the live vaccine, this is where we have seen then the question about a more serious adverse event, but still the information isn't clear whether or not that was related to the vaccine. But that's where there will be kind of the inclination, if you're going to have these types of adverse events, it tends to be associated when you have a live component. The issues that we will have to address from a nursing perspective is how we will take the vaccine once it is released from phase 3 and look to see how we will ensure storage and administration, and then how we'll communicate with our patients and others regarding safety. So when the vaccine is released, then it is called phase 4, and it would be released under an emergency use authorization. That means there would almost be a consent form that every individual, some paperwork that every receiving individual would need to sign and read and sign, that says, I understand where we are in the vaccine. I understand that we have not had millions receive the vaccine. We have had perhaps 100,000 receive the vaccine, and we are making our determinations about safety and effectiveness from that. So it gets down to the point that we will then be involved in weighing risk and benefit. And then people will always say, you know, what do you do? For me, I'm going to say, I'm going to be in line for the vaccine because I know the risk of the disease and I don't want it. I'm in the population where if there is a serious outcome, now my immune system, whether I want it to be or not, I'm not 50 anymore, I'm 60. And so I'm in that population where the high risk complications are more likely to happen. So I will be accepting the risk and hoping that I'm going to be in a priority group at first, but perhaps not. Many of you have heard about Operation Warp Speed. The CDC just released what's called the playbook, and it is available by, you can Google it and actually read the document. And the Operation Warp Speed is simply that. We're pushing forward therapeutic agents, testing agents, and vaccines. So the normal years of evaluation and development are being compressed. A lot of the work that is being done, at least with vaccines, is based upon work that it has been done slowly over many years, but now has been accelerated. But we have a very fast timeline because every day we see hundreds of people in communities and states becoming ill and individuals dying. So there is definitely pressure to bring a vaccine, if it is safe and if it is effective, to bring it to market as soon as possible. The federal government has provided billions of dollars for all of this development. And everybody is, as I'm sure, heard about the vaccine companies that have come together and kind of signed a pact to say, we don't want to bring an unsafe vaccine to market. That will kill us as a company. We don't want to do that. We want to bring a safe vaccine to market. And they are both motivated because, remember, this is a disease that impacts everybody. So it doesn't matter where you are, and it is a disease the entire world is focused on, and people are working together like never before. So when a vaccine does come to market, it will have to have demonstrated safety, and there are already some parameters that are in place that they must have demonstration of effectiveness when compared to placebo groups. Public health has a multitude of documents, and every community is working on how we will get to vaccine administration safely. Every community, and I know at the state, there is a tremendous amount of work that is going on now to try to understand what our process should be. How will we distribute first doses? How do we make sure that we can maintain in appropriate temperatures so we have no vaccine waste? How do we make sure that we have people that are knowledgeable about administration of the vaccine? And then what steps do we need to do to begin to develop a workforce? At the same time, there are a lot of therapeutics that are being released, and any of you that are working in long-term care or have students working in long-term care, there is an infusion of antibodies now that's similar to the convalescent plasma that we've read about in the news, but it doesn't have all the clotting factors and all the problems associated with the plasma infusions. These are direct antibody infusions, and they're targeting staff and patients in long-term care facilities. So Eli Lilly is working with long-term care facilities across the United States to bring that therapeutic in addition to vaccines. So tremendous amount of work. This is what the playbook looks like, the planning document, and I would really suggest that you take a look at that so you can begin to understand what is in it and how that will impact both the academic side as well as the practice side. But this is the important slide that shows in phase 1 at early release, there will be a few number of vaccines available. These may only be available through clinical trials, but as soon as the vaccine reaches then the parameters set forth by the FDA to demonstrate the primary and secondary endpoints of effectiveness and safety, everybody wants to be poised to get those vaccines out. So, for example, I mentioned Pfizer is the one that I know a little bit more about. They have the distribution even down to the packing crates and the GPS and monitoring where those crates are to be delivered so that they can immediately be available and put into use. So as soon as vaccine is delivered, it would be ready to administer in 5 hours, because that's how long we would have to take to get the vaccine, get it logged into a refrigerator or room temperature where it can thaw, and then start the administration process. Phase 1 is going to be at the bottom. 1A are paid and unpaid persons in healthcare who have the potential for direct or indirect exposure to patients or infectious material, and those who are unable to work from home. So this is frontline healthcare workers that are going to be in that 1A, and then 1B are going to be the group of other essentials and those that are at high risk for complications. So these are the targeted first groups that are part of this vaccination plan as of present. When, again, I want to show this slide back because people will ask you and ask individuals about safety. So you need to be aware of what happens during a clinical trials. We need to also be aware of our role in addressing vaccine hesitance, that people are going to be afraid. I hear this all the time. How could this vaccine be any good? It's all happened so fast. It's too new to be safe. somebody else go first and let me know how it goes for you, and then I'll make a decision. You know, a lot of questions that I'm sure you're thinking too, that we need to be looking at how we can bring some level of assurance to individuals that the first question regarding, is the vaccine going to hurt me? That has been addressed through the exploratory and then phases 1 and 2. Phase 3 now we're looking at also, is the vaccine going to help for long periods of time, beyond just a few months? Again, be able to address any issues with doubt and lack of confidence. And there's a difference between vaccine hesitance and vaccine confidence. Hesitance means I'm afraid it might hurt me. Confidence means, will it help me? So we need to be thinking and communicating that with our staff. Again, everybody is at risk for this, and we have the asymptomatic carriers. So it's very difficult for us to determine who of the priority group should go first and who doesn't. We are still continuing to learn that we must coexist with this virus, so we can't stay at home and live in our basement. We have got to get out, and we've got to figure out how we restart and how we do that safely. So our students then will be part of discussions with these community groups. Also recognizing that a large proportion of nurses especially are women, and women of childbearing years. And so protecting then our students and others, because the disease impacts every body system, including the unborn child. Safety and handling, areas where I would like for us as schools of nursing to focus, and that is, what do we need to do to train our students? How do we train them to be vaccinators so they can report to a site and they know, how do I handle a vaccine that has to be diluted? What are safe injection practices? How do I select the right needle size? How do I make sure that I have infection control practices as part of my second nature? And then how do I begin to educate the patient about not only this vaccine, but what needs to happen for a second dose?